Good morning and welcome to the Endocrine Society's news conference on endocrine-disrupting chemicals. My name is Jenny Glenn Gingery. I am the Director of Communications and Media Relations for the Society. It's my pleasure to welcome you and our speaker panel this morning. We'll be exploring the latest research on endocrine-disrupting chemicals. We're pleased to have with us Dr. Emily Hiltz, a postdoctoral fellow at the University of Texas at Austin, Dr. Chang Han of the Boston University Chobanian and Avedisian School of Medicine, and researcher Mary Bunnell of the University of Illinois Urbana-Champaign. Over the next 30 minutes or so, each speaker will present their findings, and we'll end with a Q&A session. Please note that this news conference is being live streamed, and there are a number of journalists online with us right now. Welcome to those of you who are online. Because we're broadcasting live via the web, it's really important that any remarks you make be made into a microphone. Once we start the Q&A, my colleague Colleen will be coming around with a microphone, so please make sure you speak into that, just to fully include everyone online. I would now like to invite our first speaker, Dr. Hiltz, to present.
Thank you, Jenny. I'd like to thank the organizers for inviting me today. My name is Dr. Emily Hills. I'm a postdoctoral fellow at the University of Texas at Austin, working in Dr. Andrea Gore's lab. And today I'm going to be speaking about some of the research that I've been doing, looking at the intergenerational cognitive effects of endocrine-disrupting chemical exposure in a rat model. So just to provide some brief background, endocrine-disrupting chemicals, or EDCs, are chemicals used in industry and in personal products that interfere with the normal functioning of the endocrine system. There's a very large body of research connecting these chemicals with reproductive disturbances and also with metabolic disruptions. But more recently, we're seeing neurological impairments as well. Importantly, exposure to EDCs are increasingly vulnerable during hormonally sensitive periods of development, such as during pregnancy or fetal development. And those disruptions can extend across generations via changes to the germline within the fetus that's being exposed to EDCs. This is relevant because we're witnessing a rise in cognitive disruption diagnoses. The CDC reports nearly double the number of ADHD diagnoses since the 1990s. We see similar trends for mood and eating disorders. And this has to have some kind of environmental source because such a huge rise in diagnoses cannot be coming from a simple change in genetic regulation, for example. So I mentioned these disorders because they're commonly regulated, at least in part, by brain dopamine, which is involved in the processing of rewarding information, regulates our attention, and also is involved in affective or mood processing. As EDCs can have these kind of neurodevelopmental effects via various biological mechanisms, such as disrupting hormone signaling or direct toxicological effects on the developing brain, as well as extending those effects across generations via epigenetic modifications to the genome. This means that adults, their direct offspring, and future generations are all at risk of behavioral and biological disruptions via EDC exposure. In my experiment, we exposed pregnant and nursing rats to endocrine-disrupting chemicals and then determined cognitive behavioral changes in 3 generations. Essentially, we fed an EDC, in this case, Aerochlor 1221, which is a weakly estrogenic mixture of polychlorinated biphenyls that is associated with both reproductive and neurobehavioral changes in rats. And we fed that to pregnant and nursing dams, thereby exposing the adult, her direct offspring, and germ cells within that rat that would go on to become her future grandchild. And then we assessed their cognitive behavioral changes after the fact in adulthood. The first test we measured is called a Sucrose Preference test. This is a hedonic measure meant to kind of be a mood measure in rats. They show a natural preference for sucrose, and when we see a reduction in that sucrose preference, we infer this to represent anhedonia, which is a core symptom of depression. Alternatively, if we see an increase in sucrose preference, we infer that to represent increased pleasure seeking, which you would see in something like a a substance use disorder, for example. Now let me briefly orient you to these graphs because most of the graphs I'll show today are set up this way. On the left, we have females, and on the right, we have males. On the bottom axis, we have generations. F0 is the adult being directly exposed. F1 is that fetus developing inside of her. And F2 is that future grandchild generation. And then we have our vehicle in green and our aerochlor exposure in blue. In this graph, you're seeing preference for the sucrose solution on the y-axis, and we saw no change in the overall preference for sucrose solution at the 24-hour period, but we dug a little bit deeper on this in a second cohort of animals and also measured preference over time. In these graphs, you're seeing the F1 generation, or the fetal developing rat, on the top, and the F2, or the grandchild, on the bottom. And here we saw that aerochlor exposure actually increased preference for the sucrose solution up until the 12-hour period. And this is kind of when rats are their most active. So this is when the rats are moving, drinking, feeding. Showing that rats, grandchildren of rats exposed to aerochlor 1221 have this increased pleasure-seeking behavior. Next, we measured something called conditioned orienting. This is an incentive motivational measure where rats are trained to associate a light stimulus with the delivery of a food pellet. Some rats will reliably engage in conditioned orienting responses, and I have previously shown that these types of behaviors are associated with increased impulsivity, heightened reward preference, and it's a dopamine-regulated response. So this is kind of an attentional phenotype in rats. I'll play a brief video so that you can visualize what an orienting response looks like. On the left, you'll see a rat engage in multiple orienting responses when the light comes on, and on the right, you'll see a rat exclusively engage with the food cup. Now let me play that for you. So there you can see the rat is rearing up towards the light multiple times, and the other rat is engaging with the food cup. And this is an increased incentive motivation for that light stimulus. So, while we saw no overall significant difference in the number of orienting responses displayed between males or females depending on their aerochlor exposure, if I can bring your attention to this F2 generation in males, you'll see that there is an overall shift in the population of these animals where they're showing an increase in their overall orienting. So while this isn't significant, male grandchildren of rats exposed to aerochlor may have an increased incentive motivational phenotype for rewarding stimuli. And we're still finishing out our second cohort of animals for this response. Finally, we measured attentional set shifting. This is a measure of cognitive flexibility in rats, where they're required to learn a set of responses in order to receive a reward. And then they must shift their response strategies to basically learn a new response to continue receiving rewards. What this looks like, for example, the rat is required to always nose poke to one location in order to receive food. And then once it's reliably demonstrating that response, it has to shift to this visual discrimination. Now it's always nose poking into the lit port to receive food. And the number of trials it takes to shift that response strategy is considered a measure of cognitive flexibility, or their ability to engage in new flexible responses. We didn't see any difference in the number of trials required for these animals to initially develop that response discrimination set. However, we did see an effect of the aerochlor exposure in the number of trials required to shift their response strategies. Specifically, again, in that F2 generation that was exposed as germ cells. Here we see that both the aerochlor exposed females and males required more trials to reach criterion on this behavioral shifting task, which indicates that grandchildren of rats exposed to aerochlor 1221 have reduced cognitive flexibility. Next, we wanted to dig in on some of the potential neuromechanisms of this behavior. As I mentioned, these are dopamine-regulated responses, and so the first logical step is to look at dopamine cells in the brain. Here you're seeing a slice from this experiment of dopamine immunohistochemically stained cells. These are showing tyrosine hydroxylase, which is the rate-limiting enzyme in dopamine synthesis. On the left, we have the ventral tegmental area This is an area that regulates reward motivation and attention to conditioned stimuli in the environment. And on the right, we have the substantia nigra. This is a region of the brain that is also involved in reward motivation and engaged and is also active during motor function. In the substantia nigra, we saw no difference in the number of dopamine cells present in this experiment between groups. However, in the ventral tegmental area, which is that area involved in reward motivation and the attentional processing of conditioned stimuli, we did see that males, specifically exposed to Aerochlor as adults, had a higher number of dopamine cells compared to their female-treated counterparts. And we also saw that this effect reduced over generations, such that by the F2 generation, that germ cell-exposed animal, this had returned back down to a baseline effect. So these males had reduced dopamine in the VTA by the F2 generation. We also looked at the interrelationships between this dopamine availability and some of the behaviors that some of our behavioral measures. Here, we're looking at the number of dopamine cells, specifically in the substantia nigra, and the number of errors exhibited in the set shifting task. And we found that while there was an overall predictive relationship between dopamine cell number and errors exhibited prior to learning that shifted response, there was no effect of the Aerochlor exposure. What's interesting about these graphs really is that this negative relationship, so as dopamine number decreases, the number of errors increases, is true for all generations, but the F2 generation, which was showing those Aerochlor-mediated effects, is significantly different from the adult population, meaning that this is representing some kind of separate population, although the factors that are driving that are not accounted for by the dopamine or the Aerochlor exposure, but some other mediating effect. We also saw this in terms of conditioned orienting. Here I'm only showing female data from both the substantia nigra and ventral tegmental area and those number of dopamine cells. There's a positive relationship here again, such that as dopamine cell number increases, so does the amount of conditioned orienting. And again, this F2 generation is showing a significant difference from the F0 generation, representing a different population of animals. We also measured estradiol via radioimmunoassay, and we collected that from males and females. I just want to briefly say that we saw no effect of the Aerochlor exposure on estradiol, but we did see that the serum E2, specifically in females, was a predictor of those errors in the set shifting response, and that this relationship was specific to the Aerochlor exposure. So as serum levels of estradiol went down in Aerochlor-treated female rats, they made more errors on their task, on their behavioral task. So, just to briefly summarize this work, grandchildren of rats exposed to EDCs do have an altered cognitive phenotype. They show increased pleasure seeking and reduced cognitive flexibility, and these are observed in ADHD. Where this research highlights specifically that future generations are at some of the highest risks for cognitive impairment via EDC exposure via programmed developmental changes. Our current research is exploring how these changes are occurring, and we're looking at that via gene expression profiling and exploring also the epigenetic mechanisms via which this is occurring. I'd just like to thank my lab, my co-authors, and especially my mentor, Andrea Gore. My research is funded by the National Institute of Environmental Health Sciences, which recently awarded me an F32. And I get a lot of questions about how we can avoid endocrine disruptors. So I've been working on an app, which I would enjoy speaking about if any of you are interested. Thank you very much.
Thank you, Dr. Hills. I'd now like to call Dr. Hahn to the podium.
Thanks to the organizer for having me here. So my name is Chen Han. I come from Boston University Chopin and Aviation School of Medicine. I'm delighted to represent the authors from our research group to discuss with everyone our research findings titled Association Between Mixed Dioxin Exposure and Thyroid Dysfunction in U.S. Adults. Firstly, I would like to present a brief summary of our conclusion from our research. Our study provides new insights into the complex associations between dioxin and thyroid dysfunction in U.S. adults. Using linear regression model, we find significant association between exposure to 10 different individual dioxin and increased TSH levels. The results obtained from multi-pollutant models using WQS and BKMR model demonstrate that co-exposure to multiple dioxin was associated with an increased prevalence of hypothyroidism among adults. As we know, thyroid dysfunction is a condition in which the thyroid produces too much or too little thyroid hormone, with a prevalence up to 11% worldwide. According to American Thyroid Association, more than 12% of U.S. population will develop a thyroid disorder during their lifetime. In terms of the impact of thyroid diseases, hyperthyroidism is associated with increased risk for cardiac complication, while those with hypothyroidism are at increased risk for dyslipidemia, hypertension, heart failure. Hypothyroidism during pregnancy is also associated with adverse neurodevelopmental outcomes in offsprings. Therefore, it is very important to identify the causes of thyroid dysfunction for its prevention and diagnosis. Many factors are associated with the risk of thyroid dysfunction. Traditionally, thyroid autoantibodies, iodine excess, and iodine deficiency, these are all well-known causes of thyroid dysfunction. However, they only account for 20 to 70% of cases. So what other potential reasons could lead to thyroid dysfunction? Recently, interest has focused on the potential impact of exposure to environmental chemicals on thyroid function. Our research group is particularly interested in the impact of dioxin on thyroid function. Dioxin are a group of highly toxic chemical compounds known for their environmental persistence and which may cause a range of adverse health effects. Dioxin can be divided into three groups. They are PCDD, PCDF, and PCBs. They are primarily produced as a result of industrial process, such as waste incineration and the production of certain herbicides and pesticides. Dioxin are highly resistant to natural degradation and accumulates in the food chain, leading to increased exposure level in humans. High dioxin exposure has been associated with serious health problems, including cancer, damage to the immune and reproductive systems, and developmental disorders in fetuses and children. In this study, we used and compared 3 different statistical models to investigate the combined effects of environmental dioxin exposure on thyroid dysfunction using data from enhanced participants. Each of these methods has its own strengths and weakness. By comparing the results of the 3 models, we aim to provide new insights into the relationship between exposure to dioxin mixture and thyroid dysfunction. Study design and method. The ENHANCE is a large population-based survey conducted by CDC and NCHS. We use data from 2007 and 2010 ENHANCE cycles. Participants aged 12 years and older who had completed measurements of thyroid function and dioxin were included in our final analysis. After excluding those without data of thyroid function, excluding those without data of TSH, and those who are pregnant, and exclude those who are taking medication which may have impact on thyroid function, and exclude those without data of important covariates, a total of 2,818 participants enrolled in our final analysis. In linear regression model, the level of dioxin is divided into 4 quantiles. All models were adjusted for age, sex, race, body mass index, smoking, and urinary iodine. We identified a total of 10 individual dioxin which are positively associated with increased TSH. There are 5 in this slide which show a positive association with increased TSH. There are another 5 in this slide which also showed a positive association with increased TSH. Then we adopt WQS regression model. Based on the results of our linear regression model, we calculate the positive direction of effect of combined exposure to the dioxin on high TSH. Based on the previous studies, we adjust age, sex, race, BMI, smoking, and urinary iodine in WQS regression model. WQS regression helps us to model the combined effect of dioxin exposure on increased TSH and relative contribution of each dioxin exposure to the effect of exposure index on the outcome. Our results indicate that the combined effects of dioxin is positively associated with increased TSH. Individually, 1, 2, 3, 7 PNCDD makes the greatest contribution among all dioxins. We also use BKMR model to estimate the individual and joint health effects of exposure mixture and realize potential nonlinear exposure response associations. Our results indicate that individual dioxin did not show a positive correlation in the BKMR model. However, the combined exposure of dioxin from 30th percentile to 65th percentile did have a positive association with increased TSH. I would like to express my sincere gratitude to my mentors, Dr. Elizabeth Pierce and Dr. San Li, and my colleague Carol Peng for their invaluable support throughout the course of this research. That's all. Thank you.
Thank you so much, Dr. Hahn. I'd now like to invite Mary Bunnell to join us.
Good morning. My name is Mary Bunnell. I'm a graduate student at the University of Illinois at Urbana-Champaign in the co-laboratory. Thank you for the opportunity to present this work today. This is a project that is focused on prenatal phthalate exposure and sex differences in response to these chemicals. I have nothing to disclose. So why did we choose to study phthalates? These chemicals are very widely used in many products that humans are exposed to on a daily basis. They're man-made chemicals, and the reason they're added to products is as a solvent or to make plastics more flexible, really for textural purposes. And as you can see, they're found in things from vinyl, which is used in our homes, in our flooring, cosmetics and personal care products that we're applying directly to the skin, plastic products where we store our food and make children's toys from, and medical tubing. These chemicals are endocrine disruptors, and how these can impact human health is when we are exposed to them, they mimic or block the actions of sex hormones in the body, and this disturbs the normal function of our endocrine systems. Concerningly, there are multiple routes in which we can be exposed to these chemicals, by skin absorption, inhalation, ingestion, and intravenously. And today I'll be talking about prenatal exposure because phthalates do have the ability to cross the placenta and reach the developing fetus. And why did we study sex differences? So we have a knowledge gap today in biomedical research, which is due to historically the majority of biomedical research has been conducted using male subjects, male cell lines, or animals. As a result, we do not have a full understanding of how sex influences many important biological processes. So in my study today, we used animal model. So when I say sex differences, I'll be referring to biological males and females, in which we define their sex according to their anatomy, physiology, genetics, and hormones. So according to phthalate exposure, there are known sex differences in the health risks that come with being exposed to these chemicals. Men are more likely to have problems with sex organ development, decreased sperm number and quality, and decreased testosterone levels. We know a lot more about the effect of the phthalates on males. It could be due to the fact that many of these ailments are just more obvious in men. Whereas in women, they would have hormonal imbalance, increased risk for pregnancy and birth complications, earlier puberty and menopause, and breast cancer and endometriosis. And endocrine puberties are really important in establishing sex differences. And so we chose to study this process in hopes to understand a little bit more about the reason why we have sex-dependent impacts from these chemicals. So endocrine puberties are times when key hormones are elevated. These are different in males and females. In the males, it's testosterone. In females, it's estrogen. Estradiol is the primary form of estrogen. And these raises in the sex hormone levels are very important for reproductive development and establishing sex differences. So you'll see on the left, at classical puberty, you would all be familiar with, we see physical effects such as the ability to reproduce. But prior to that, there's an endocrine puberty during the fetal period and an infantile endocrine puberty immediately after birth, where males have very high level of testosterone and females have a high level of estradiol. And these are very important events in laying the foundation for sex differences and their future reproductive development. And so because of this, children that have not gone through puberty yet are more susceptible to phthalate exposure. In our study, we used the mouse as an animal model because they also go through the endocrine puberties in the way that humans do. So we asked the question, after being prenatally exposed to phthalates, did the mice, male and female mice, respond differently during these critical times? To do so, we used an environmentally relevant dose of phthalates, which was orally dosed to pregnant female mice. And during their fetal and infantile endocrine puberty of the offspring, we measured their hormone levels, and then we collected their liver as well as other tissues so that we could investigate a potential reason for why these changes that we would see might be happening. So during their fetal endocrine puberty in the males, we saw a slightly increased level of testosterone. However, in the females, their testosterone levels were decreased at this time. And during infantile endocrine puberty, if you remember the graph I showed previously, the males have a surge of testosterone. So hour 1, 3, and 6 after birth, that level of testosterone is on the decline, as you can see. And in the males exposed to phthalates, this occurred normally. There was no difference between the control animals and those that were exposed. However, the females at hour 6 had significantly lower amount of estradiol. And this could indicate to us that they are clearing this hormone out at a faster rate. And in addition, at fetal endocrine puberty, the females also had a lower level of testosterone. So we asked the question of, how does the liver's role in the reproductive system come into play here? The liver actually has a very important role in regulating your hormonal levels. They will biologically inactivate or remove these hormones to be excreted from the body. It also has a high level of sex-dependent gene expression and different function between males and females. In addition to those things, it undergoes a drastic development and changes in function around the times of these fetal and neonatal endocrine puberties that we are studying. We use the technology of RNA sequencing to get a snapshot idea of the changes in gene expression in the liver at these time points. And we wanted to see if there's a difference in the response between males and females. And what we find is that the females have upregulated genes that are involved in metabolizing these hormones. And this may indicate to us that they're metabolizing them and removing them at a faster rate. And this is in line with the hormone data that we previously saw. Histology is the microscopic structure of tissues, and so we use this to evaluate on the microscopic level whether there's any changes in the morphology of the liver. And you're looking at liver cells under the microscope right now. Of note from this slide, we did not see any drastic difference between the groups, and that tells us that this change is likely not due to any major change in their morphology. Then we stained the estrogen receptor 1 protein in the female infant liver, and you can see where estrogen receptor 1 is expressed, where these cells are stained brown. And estrogen receptor 1 is necessary for estrogen's effects to happen on the liver, and it goes into many different functions of the liver. It regulates other metabolic functions as well as contributing to sex differences. And we found that in the phthalate-exposed females, they had a lower expression of this estrogen receptor 1. In summary, when we expose pregnant mice to phthalates, their offspring, during critical windows of development, the females appear to have altered metabolic gene expression in the liver, which is leading to an abnormally rapid removal of hormones during a critical time of development. Due to this occurring at a critical time during their endocrine puberty, it could put them at more risk for abnormal reproductive development and function. So in conclusion, the females appeared to be more sensitive to males to the same chemical exposure during their fetal and infantile endocrine puberty. And because these are critical windows of development, that altered liver metabolism could possibly explain some of the adverse effects of phthalate exposure on women's health. And finally, I'd like to acknowledge my mentors, my lab mates, and collaborators who made this work possible. Thank you.
Thank you so much, Ms. Bunnell. We're now ready to open the question and answer period.
Hi, Ed Sussman with MedPage Today. Dr. Hahn, I've got a few questions regarding your study. Is there a threshold of dioxin exposure that can cause high TSH levels?
Because previously, based on previous study, especially in 2018, European Food Safety Authority, they said that there's no strong indicate that dioxin exposure is strongly related with thyroid function, because previous study, they are relatively has small sample size, so they cannot give us a strong evidence. So based on previous study, we do not have a threshold for that. So we are trying to see whether there could be a threshold. So that's why we divided our dioxin into 4 quantile. So usually, individually, the 4th quantile may have a positive association with higher prevalence of thyroid disease.
Should people with documented thyroid disorders be tested for dioxin levels?
Ideally, I think we should be cautious about that, but it's hard to control that because 90% of dioxin for human beings, in people in the US, the dioxin exposure, 90% are from food exposure in the food chain, so it's hard to control it.
Is there some way to remove dioxins from the body once they're there?
Remove it from body, it could be hard. So what we can do is try our best is to remove it from what we intake. For example, dioxin is mainly accumulate in fat tissue. So if we eat less fat tissue, eat more lean meat, that could be good for us to take less dioxin.
Thank you.
Thank you.
Great, thank you. Anyone else in the room have a question? I had one question for Ms. Bunnell. How can this mouse study inform the way we think about exposure to EDCs and their impact on newborns?
Yes, thank you for your question. So obviously we cannot use humans and dose them with phthalates and see how they respond to this exposure. So phthalates, mice make a great model organism to study this process, and we're able to use them to investigate the endocrine puberty because mice, it's also important for their development in the same way that it is in humans. And so as opposed to studying humans, we do use mice as an animal model, and we look at what happens in the mice. We look at human data that we see from epidemiological studies, and we want to consider the results of the animal studies and what we know in humans and try to get a full big picture understanding of what's happening.
Great, thank you so much. One question too for Ms. Hills. What can be done to reduce PCB exposure?
PCBs were banned in the 1970s, but they remain present in our food chain, air and water, because of their resistance to biodegradation. So this is a body burden that we're all carrying. They're also still used in industries in other parts of the world. So one thing would be reducing their use in industry in other parts of the world because they are via migration continually, like we're continually exposed to them. But I think alternatively, we need to understand what they're doing to us so that we can kind of develop, at this point after the fact, types of treatments to deal with the outcomes.
Great, thank you so much. Do we have any questions online? Okay.
Dr. Hills, can you share a little bit more about the app you're developing? I know you won a grant award to develop it.
Yeah, through the Office of Women's Health and the Human Health Services, I won, along with a couple of other people, a EDC Innovator Award, which is used for increasing Black women's awareness of endocrine-disrupting chemicals, specifically in personal care products. Personal care products like the lotions we use, the soaps, have a lot of EDCs in them, unfortunately. And the way that the app works is you can take a picture of an ingredients label, and it references a database that I built on top of some other researchers. And it will basically give you a feedback and tell you if any of the chemicals detected on the label are known EDCs, as well as the health outcomes associated with that. And then it'll also give you the exact number of literature papers, like science papers, associated with both the overall EDC and the specific health outcomes, so that you can kind of decide for yourself if that's a threshold of evidence for you. So,
Fascinating. We'll be watching for that.
thank you. It's very much, I included it. It's not very pretty right now, but it does work.
Great.
Thank you.
Well, thank you again to all of our speakers for joining us today. This is such an important issue and certainly a priority for the society, and we're glad to have you here to tell us more about the latest science. As a reminder, the recordings of these news conferences will be available on our website and our YouTube channel in the coming days. If you have additional questions or would like to set up an interview with one of our speakers, all you have to do is email us at media@endocrine.org. Thank you so much and enjoy your day at Endo 2023.